Good morning, BRAOU, Ambedkar University, Biostatistics students. I am Dr. G. Lavanya. Today I am going to explain biostatistics. The content is already I started the class. The content is you have three units. Unit 1, purpose and structure of biological assay, types of biological assays. Direct assays, ratio estimates, asymptotic distributions, Feller's theorem, and regression approach to estimate those response and relationships. So already I started the class. The topic is purpose and structure of biological assays, types of biological assays. Already I started the class. Purpose and biological assays, types of biological assays. Okay, what does mean biological assay? I explained in previous class. Who invented biological assay? That also I explained. And today, some of the leftover topics are there that I want to explain. The types of biological assays, direct and indirect assays also I explained. Okay, this is the information how the example problem is lies in mean percentage standard deviation. Okay, that is okay. Next, and this is the topic I would like to explain, that is direct assess ratio estimates. Direct assess ratio estimate. So in this, the main objective is components of biological assay, basic process and directed assays, modeled for direct assays and analytical dilution assays, possible solution for analysis assays, ratio estimates, definition. Biological assays are methods for estimating the potency or strength of agent or stimulus by utilizing the response or effect or reaction caused by its application to biological material or experimental living subjects. 6 aspirin tablets can be fatal to a child. Okay. Certain dose of a lethal drug can kill a cat. So this is some examples. So from Webster International Dictionary, we can say biological assay is the estimation of the strength of a drug by comparing its effect on biological material, an animal or animal tissue with those of a standard product. In other words, we usually can only estimate relative potency of an agent, not its potency. Not its potency, only relative potency we will estimate. So, components of a biopsy, the subject is usually an animal, a human tissue, or bacterial culture. The agent is usually a drug, a clinical chemical. So here, so what does mean? The response is usually a change in particular characteristics or even the death of a subject. Response can be binary or measured on continuous scale. There are deterministic or non-stochastic tests, but they are not subject to statistical analysis. So they are not our targets. C is stochastic if the relative potency is influenced by factors other than the preparations. That is, extraneous factors which cannot be completely controlled and explained. That means the factors which happens but which we cannot see in our naked eye. So that is the response is subjected to a random error. Here, dose or the response is the so depending on the design. So next basic process. First acoustic assays, our only targets, we refer to the relation between stimulus level and the response it produces as a regression model. A test preparation of stimulus having an unknown potency is assayed to find the response. We find the dose of the standard preparation which produces the same response that by test preparation. Direct assays, indirect assays, they are both stochastic. Stochastic process means here the present time is dependent on its immediate or next trial. That is called stochastic process. So here the direct assays, the dose of the standard and preparation are directly measured for until an event of interest. Response is fixed binary, dose is random. Okay. So when an event of interest occurs, the deaths of the subject and the variable of the interest is the dose required to product that response event for each subject. Means whenever we can give the dose, immediately the response occurs. So whether it is positive, means no problem, then we can say it is okay. If there is arise any problems in any symptoms, then we say the dose is not worked. So the value is called individual effect of dose. That is called IED. So we can increase the dose until the heartbeat of an animal is to get IED. So that is, so if we increase, then what the effect? That also we can note. So, okay. Now, typical experiment. Exposed subjects, for example, you take animals, are randomly divided into two subgroups, and then IED means what? Individual effect of dose of standard preparation is measured in each subject of group 1. Individual effect of dose of the test or unknown preparation is measured in each subject is 2. So one is known group, one is another unknown group. So these are the two groups. So from this, the aim is to estimate the relative comparativity potency, that is ratio of concentrations of the test relative to standard to produce the same biological effect event. Keep in mind that the concentration and the dose are inversely proportional when concentration is high. We need a smaller dose to reach the same response. In other words, if we define the relative potency or the ratio of concentrations of the test to standard as the ratio of doses of that standard test were equal to rho equal to dou S by dou R. So if we give the small dose, there may be some. If we increase the dose, there may be some. So this efficiency we can calculate, that we can define rho equal to dose S suffix dou R. When the relative potency is greater than 1, the test preparation is stronger. We know that the correlation coefficient is lies -1 to +1. So +1 means it is a stronger. If rho greater than 1, potency means here ratio value is greater than 1. The test preparation is stronger, not the rank ma. Here the that is greater than 1. Actually, it lies only plus or minus 1 only. Here ratio, the ratio, not the correlation coefficient limits. If the ratio is greater than 1, that is called stranger. We need larger dose of the test in order to produce the same response, vice versa. So pairs of doses that gives the same response are termed equipotent, meaning same strength. Equipotent means same strength. Means here simple we can group one, group two. One is known group, one is unknown group. We are giving the dose to each group. There may be if some small dose, some effect. Dose increase, some effect. So we need to calculate the ratio. So that value is greater than 1. You assume, you give the conclusion inference that is the stronger. And also we can find out equipotent of those groups. Okay. So data are very simple in two independent samples. The type you usually have for two sample test T. You know that small sample T test, the difference of mean of small samples with the size is 25. Okay, ma? So with Gaussian test, this is non-parametric test, but we will not compare them using a test of significance. We want to estimate relative potency. Recall that we define the relative potency as the ratio of concentrations of the test to standard, or the ratio of doses of the standard to test. Point estimate is easy. The more difficult part is its precision and confidential interval. We would need a statistical model. For students of statistics or biostatistics, the only new feature is learning to deal with the ratio of sample means instead of the usual differences of sample means. For example, with exact variance, we usually approximate it by using delta method. Next one is indirect assess. What does mean indirect assess? The dose of the standard and test preparations are applied and observe the response that each dose products. For example, here we measure the tension in a tissue or the hormone level or the blood sugar content. For each subject, the dose is fixed in advance. The variable of interest is not the dose but the response it produces in each subject. The response could be by yes. So here we measure the tensions in a tissue or the hormone level or the blood sugar content. For each subject, the dose is fixed in advance. The variable of interest is not dose but the response it produces in each subject. The response could be binary or continuous. Statistically, indirect assays are more interesting and of course, all are difficult. It's more difficult. Indirect assays, the dose is fixed and the response is random. See here, dose is fixed, response is random. Here we are not changing the dose. Same dose we are giving and we are finding the response. That is the indirect assay. Please keep in mind. Okay. So now here that response could be a measurement or the occurrence of an event, whereas the response in direct assay is always binary. The occurrence of an event. What does mean binary? We have two situations, that is positive or negative. So that we have to note. Next one is measurement scale. Depending on the measurement scale for the response of indirect assays, we have one is quantal assays, where the response is binary, whether event occurs or not. That is, binary means it occurs, it not occurs. Anyone answer? So let the death of the subject occurs. Next one is quantitative assays, where the measurements for the response are on a continuous scale. The common indirect assay is usually one in which the ratio of equipment dose is estimated from curves relating quantitative responses and doses for the two preparations. The shape of these curves further divides quantitative indirect assays into one is parallel line assays are those in which the response is linearly related to the big dose. Next one is slope ratio assays are those in which the response is linearly related to dose itself. Indirect assays are also divided into analytic function or comparative function. So here, analytical dilution assays are such that the test and standard preparations behaved as though they are identical, same constituents, exact for the concentration. Next one is comparative assays. The two preparations are not the same. For example, the concentration of one protein is estimated by using a different protein as standard. For analytical dilution assays, the only difference is concentration. The constant relative potency is the reciprocal of the dilution factor. In other words, its extent solution is global. That is, a solution always exists and is constant. For comparative dilution assays, the response producing constituents in the two preparations were only qualitative and quantitative. So, value of the space relative potency may not be constant. In other words, its existence is local statistical analysis are mostly the same. However, the technique unless we know the chemical or biological system well. Most of the times, it is impossible to tell an analytical dilution assays from a comparative assay from the resulting data. The exception is perhaps that assays. So here one of the model for direct assessment. Here you have to take x1 etc. x entity identical dilution independently identically distributed variables with the normal where mean is mu variance sigma square. So another one is independently identically distribution where normal mu s and sigma square. The suffix or suffix s is the two sample observations, independent sample observations. Here we calculate rho equal to mu s by mu r. The following results are well known for normal distribution where r equal to x bar s by x bar r. Where expression of r equal to rho, variance of r equal to sigma square by mu r square in bracket 1 by ns plus rho square by nr n to the power of 1 by 2 into r minus rho equal to normal mean zero variance is this. The error propagation we have by using delta method, that is this. This is the standard error. You know that standard error means sigma square sigma by root n, okay, or sigma square by n. So where s square p is, you know that formula pooled variance, that is ns minus 1 into s square s plus nr minus 1 s square r by ns plus nr minus 2. So here the exact variance is approximated using the delta method. The you also. Thank you. The variance of estimated relative potency R can easily obtained approximately, but the normal distribution for R ratio of sample means fit poorly. So here, especially when the sample size are often rather small. So here what we observed, but the normal distribution for R, the ratio of sample means may fit very poorly, especially when the sample sizes are often rather small. Next one is consider the normal distribution of identical distribution, independently identical distribution of test doses with mean mu variance sigma square. This is the one set of random numbers where normally distributed, its mean is mu variance sigma square. Another set of random variables also independently identically distributed with the mean is mu variance is sigma square. Okay, so now we assume that F ratio, the ratio of independent sample the ratio of independent chi-square variate is called the F distribution. Here S square S by S square T, which distributed of n minus ns minus 1 or nr minus 1 under null hypothesis. So analytical dilution assays. Analytical dilution assays are those for which the test and the standard preparation behaved as though they are identical except for the concentration, that is x is equal to rho xr, where variance of x is not equal to variance of xr, where rho not equal to 1, the cases and we are interested in, we compare to example standard and test. And here total standard. So here one of the tests, that is standard measurements and test measurements. Means standard means if we have some temperature, our body temperature is this. So that actual value, room temperature is this, some 24, 25. So whatever, if the climate change and place change, we can test, we can get other temperature. So that means standard and test. So total mean and variance. So here, possible solution for analytical dilution essays, where x is equal to rho xr, where x is equal to log rho plus log xr. So here, variance estimate can be pulled to have more precise estimation. Relative potency is based on the anti-log of the difference of means rather than the ratio. So easier procedure where Kepler equal to exponential exp zs minus zr, where confidential interval. So that is 95% confidential interval is exp zs minus zr plus or minus t 0.975 under root of 1 by this plus 1 by nr. So here standard and test. We need to test. We can apply the test where mean is this, variance is this. Then you will get standard error of this is, we will get the value under 95% confident interval. The value is r plus or minus t sigma standard error r, where r plus or minus 1.96. This is a tabular value standard error r. Next 1.1 r is 1.18 plus or minus 1.96 is the 975 confidence limit, and expectation of r, here we calculated mean by that formula, where here given the expectation of r, that formula here we can substitute, we will get the value here. So another example, standard and test, total mean variance. So here capital R equal to EXP ZS minus ZR. So EXP Z bar S and then Z bar and mu minus sigma by root n, a value. Mu minus sigma by root n. So then you substitute, you will get this. So next, check your progress. So with competitions of biological, write the competitions of biological, components of biological assay. Write brief analytical dilution assays. That the basic process and direct assays. Next one is ratio estimates, where R equal to mu R by mu S, where that is Ty by Tx. There is summation yi by summation xi. The ratio estimated is statistical parameter and is defined to the ratio of means of two random variables. Ratio estimates are biased and corrections must be made when they are used in experiment or survey work. And the problem of interest involves two random variables x and y, particularly ratio of two means equally the ratio of their totals. So that is 1920 population, 1930 population. Here two groups. One example would where YI is the population in the year 1930 and XI is the population in the year 1920 per city per thousand. The plot shows the population 49 is the large cities for 2 years in question. So next one is expectation and variance of ratio estimation. Let capital X equal to mu plus error epsilon. So where linearity is yi is v mu plus eta. Where x bar equal to mu plus epsilon. Thus y bar equal to v plus eta. So then eta bar equal to 1 by n2 eta summation ni, where error bar equal to 1 by n summation error I, where ratio estimate y bar by x bar equal to v plus eta bar mu plus epsilon bar, that is v into 1 plus eta by v, 1 plus epsilon by mu. So here we can get expectation of y bar by x bar is x and y are independent, v by mu 1 plus sigma square n mu square, where variance is sigma square 1 by n mu square plus v by mu whole square n mu square. So that is epsilon bar by mu less than 1. So here compute your answer with the one of the given. So write briefly ratio estimates. So here, what are the components of bioassay? What are the biological assay? Typical experiment in bioassay. What is the analytical distribution assays? Explain. What are the advantages of doing analysis or log dosages? Discuss ratio estimates or dilution assays. So here, basic process of bioassays, analytical dilution assays, ratio estimates. So here, component of a bioassay. The subject is usually an animal, a human tissue, or a bacteria culture. The agent is usually a drug chemical. So these are the answers already given the first answer and second answer and third answer and fourth answer. So please get through all these answers, very, very important. My dear students, please read only these questions and write in exams well. Okay. So if you are registered as a BSc biostatistics students, you must take these notes and read. If any query, any doubt, ask me. I'll explain. Okay. So these are the short answer questions. What is the typical experiment in the bioassay? What is the analytical dilution assays? What are the measurement scale in of bioassays? So here long answers. What is model for direct assays? Discuss possible solution for analytical dilution assays. Explain result for dilution assays with two examples for any considered data. So discuss ratio estimates for dilution assays. So here next topic is unit 3. So before that, the 1 and 2 units are very, very important for you, and just I recall these 2 units what I explained. The students, the first unit is based on purposoid structure of biological assays, types of biological assays. The unit 1 is types of biological assays. Okay, so these are the objectives of the studies, history of the studies, and examples are given. So please, what is bioassays? What example might given, and these are very, very important. Okay, next one is purpose of bioassay. This and what are the types of bioassays? Please read through these topics. Okay. Next one is the data is given. What is the normal score? What is the test score? And what are the confidential limits of the test? And that also very, very important. Their students, What are the short answer questions, model questions? Please read through each and every one. Okay. So next unit is direct assess and ratio estimates. Before going to this direct assess and ratio estimate, how to calculate ratios is very very important. And here, here, examples of these biological assays. So, what is mean by biological assay? The main objective is what? Of direct assays and indirect assays. When we call direct assays, when we call indirect assays. So, these are very, very important. And next one is some problems are given. The standard values and test values. And from this, you have to find out all these. Okay. So next one is some example problems is given. Okay, then some check progress is also given. Okay, next one is the unit is 3. Here we will explain about what is a unit 3. Asymptotic distributions, Feller's theorem, and regression approach to estimate dose response and relationships. You know that distributions play very important role in statistical theory. What does mean distribution? A characterization of random variable is called the distribution. For example, random variable. How can we identify that is a random variable? In real world phenomenon, many experiments take place. Many experiments takes place. The outputs are which is continuous or binary or fixed. Okay. So like that, the where distributions are characterized according to the random variable. So a random variable plays very important role in distribution theory. Okay, we assign the values of an experiment with the help of random variables. Okay, so which is small sample and large sample, and which is characterized as binary or univariate or bivariate or multivariate. So here the main objective of these asymptotic distributions, and next one is Fellow's theorem. So here the main objective is concept is asymptotic distributions and ratio of the distributions and Fellow's theorem. So here, asymptotic theory to derive joint distributions of the estimators for parameter in a model. So asymptotic distribution is a distribution we obtain by letting the time origin sample size go to infinity. We can simply the analysis by doing so. We know that some terms converges to zero the limit, but we may also have sample error. Hopefully, hopefully, when the sample size large enough, the error becomes small and we can have a satisfactory approximation to the true or exact distribution. The reason that we use asymptotic distribution instead of exact distribution is that the exact finite sample distribution in many cases are too complicated to derive. Even for the Gaussian process, therefore, we use asymptotic distributions as alternatives. Method to obtain a limiting distribution. Stabilizing transformation transform x alpha to makes you the moments do not depend on n. So here steps. Multiply the sample statistics x by n such that the limiting distribution of nx is has a finite non-zero variance. Then transform xn to make sure the mean does not depend on n. Either the mean of sample has a limiting variance equal to 0 since variance of mean x bar is sigma square by n. So here the mean is does not depend on sample size. So this formula is very, very important. Please go through this and 1 minute. Please recall what I taught today. Okay, and tomorrow I'll start one more. And here this is enough. Today, what asymptotic distributions? Here the main one is the mean of sample. The mean is does not depend on n. The mean does not depend on sample size. Only when sample size is increases, we get error, small error. How? How the sample size increases, small error? Can you give any example? Can you give example? If you take same coin mark, first time you can toss. Same coin you repeated 1,000 or 500 times, then the coin is changed the shape of that. When you go on repeating the same coin, same trial, increase the trials, then you will get probably same answers. Same answers. So as n increases, the error is small. As n increases, the error is small. So, for example, in a company, the products is preparing. First, you can con defects. Second, you can get more defects. Third, you note down the defects. When same is repeated in many times, when defects are less, means the product is preparing, the machine is habituated the cycle. So we didn't get any mistakes. Like మనం ఫస్ట్ చదవాలంటే కష్టం. అదేవిధంగా గంటలు గంటలు తరబడి చదివితే ఆ సబ్జెక్ట్ అర్థమైంది అనుకోండి, మనం ఇంకొకరు పిలిచినా కానీ లేవం. సో ఇట్లా. సో యాజ్ ద శాంపిల్ సైజ్ ఇంక్రీజెస్ విల్ గెట్ ద ఎర్రర్ స్మాల్. సో దట్ ఈస్ ప్లీజ్ నోట్ డౌన్. And tomorrow I'll explain other one, and I'm sharing the notes, and please go through the notes once again. This is very, very important, the short answer questions, and this question is also very, very important, and this please note down. Okay. You have your time now? Okay, I'll try to explain one more topic. No, no, no. I will explain one question because it's very, very important in your exams, no? So we'll go for the answers what you have. The question is what is biostatistics? So where the answer is, so here what is the biostatistics? Some may ask the question. Then what is the biostatistics? Please tell me. So here some definitions. What is biostatistics? What is bio say? A statistician can make the following contributions: advise on general statistical principles underlining the same method, devise a good experimental design that gives the most useful and reliable results. Analyze the data, making use of all the evidence on potency. Here, design consists of choosing the number of levels of doses at which each preparation is to be tested, the number of subjects to be used to each dose, the method of allocating subjects to doses, the order in which subjects under each dose should be treated and measured, and other aspects of the experiment. Needless to emphasize that sound design is as important as the statistical analysis of data. So, biological assays are methods for estimating the potency of a drug or material by utilizing the reaction caused by its application to experimental subjects that are living. For example, how do pharmacists know that six aspirin tablets can be fatal to a child? So, qualitative assessments of material do not pose any great problems. Quantitative assays of material are our main concern. Okay, so a study of the effects of different samples of insulin and the blood sugar of rabbits is not necessarily a binary problem. However, it will be if the investigator is interested in the estimation of the potencies of the samples on a scale of standard units of insulin. Okay, so these are very important. And the next example is a study of the response of potatoes to various phosphate fertilizers would not be a bioassay. So here see, this example is not bioassay. Why? The response of potatoes to various phosphate fertilizers means if we give the different mm, fertilizers to the plant, so the yield of these potatoes, so the there may be it's not we can call it is a not a bioassay. So it is a biosay if one is interested in using the yield of potatoes is and assessing the potency of a natural rock phosphate, that is. So first there is a physical quantity like the weight of an animal which we call factor X whose levels or doses can be controlled. The effect of the factor X will be called response. The relation between the dose and the response will be described by means of a graph or an algebraic equation. So biostatistics is application of statistics in several topics. It relates topics in biology, medicine, because nowadays in medicine clinical trials are there a lot of data analysis means they have we get full collection of data there, so you need to have it in pharmacy when you are looking at new drug, when you are comparing drugs in public health science, you are looking at disease or progression. So that is. The second is agriculture. When you are coming up with the new strains, new plants, you are comparing old plants and old yield with new plants and new yield. See, nowadays we have seen there are in agriculture and plantation, there are different cultivation methods are have come in across. So here, fishery, when you are doing a lot of changes to a population of fish and you are trying to look at their growth pattern as well as a yield. Okay. Third, ecological forecasting. Again, changes in climate. In climate conditions, temperature of the sea, and so on actually, and then sequence analysis. We are also going to look at some problems there. System biologically, gene networking, bioinformatics, biology, are dealing with the large amount of for data. The sequence of genes, you are comparing two gene sequences and trying to say whether there is any statistical significant difference between them. So then based on that, you are going to analyze the pathways and then compare the performance or the mechanism of action of population genetics. You are looking at different types of population, how the diseases are prevalent in certain populations. Then, what are the habits of certain populations? What are the amount of data? And you are making a comparison between one data and the another data set. So that is data set could be very old and the other one will be new. So you might get the difference of old and new. So here in statistics we can analyze as agriculture, as a pharmacist, as different types of comparativity that we can find out. So okay, by using this statistics is very much interested in biological theory, pharmaceutical theory. Before test only, the drug is can come outside. After a lot of preparations, after a lot of tests, then only drug cured particular disease means it tested by small rats or animals, then human, they will test on human body also. So after it only the drug comes out. Okay, so this is a very important facts of bioassay. You might have to know about it. So as a biostatic students, you must learn the questions here takes place, that is explain the optimal sampling strategy. Next, polycotomous quantal response. Next, Blum's conditions, parametric estimation, Robbins monoprocess, the concept of UP and John rule, quantal response function. Next, long answers, the concepts of polychotomous quantal response. Briefly, the concept of Robbins Monroe process and Robbins procedure, Monroe procedure. Next, linear approximation, asymptotic normality, and concept of Blum's condition. So these are very important. Parametric estimate with the example. Briefly, the UP and down rule in the estimation of point in the quantal. Next, modified UP and down method. So next one, already I explained concept of gene and genotype, Mendel and Wing loss, estimation of allele frequency, the dominant, codominant cases, multiple alleles. So these are very important. And the next one is gene and genotypes. The main objective of genotype is know the importance of statistical genetics, concept of genes and genotypes, allele frequency, relative frequency of alleles and genotype. So here some statistical genetics. So we have population genetics and also have characteristics of genes, concept of genes also we learn, concept of genotypes. So what does mean genotypes? You know that in a broad sense the term genotype refers to the genetic makeup of an organism. Genetic makeup of an organism. Okay? So, in other words, it describes an organ's complete set of genes. An organ's complete set of genes. In a more narrow sense, the term can be used to refer to the alleles or variant forms of gene that are carried by an organism. Humans are diploid organisms, which means that they have two alleles at each genetic position or locus with one allele inherited from each parent. Each pair of alleles represents the genotype of a specified gene. Example in a sweet pea plants, The gene for the color of a flower has two alleles. One allele code for purple flowers and is represented by uppercase letter F, whereas the second course for white flowers is represented by the lowercase letter f. A varied population of sweet pea plants could therefore feature three possible genotypes at this locus F, 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 or F, F. Each plant's genotype contributions to its phenotype with in this case in the outward appearance of its flowers a particular genotype is described as homogeneous if it features two identical alleles and as heterogeneous if two alleles are different. The process of differ determining genotype is called genotyping. Here see capital F, homogeneous, capital F, small f, heterogeneous, 2 small f is homogeneous. Okay, so like this, each and every point is given. Please go through this one example and note down. Okay. Here one more example is given and please try to answer these questions. I am giving some time. The questions are here. So these answers. Next one, this check your progress model answers. So already you have some questions, no? So this next short answers. Next long answers. కూర్చో. ఏ క్లాసు? పొద్దున్న నుంచి ఏం చెప్తా? So these are the long questions that is explain the concept of Hardy-Weinberg equilibrium. So these all five questions, students, please practice all these questions. You will definitely pass. I request all the biostatistics BSc students, those who are registered in BRAOU, please get through and take the already sirs will give books to you. Just please read, and whenever your exam before 1 month onwards, please you practice, you will definitely pass. I hope you will pass. This is my humble request, and please read. So here this some questions. Each unit they have given the long answers and small some answers and short answers. Here, whatever the questions are there, you first get it the answer, then practice. Get it the answer and then practice. Please go through these notes. Nothing. And this is the one paper. We will check first 2 units if we have any answers, any questions or come in this paper. Define direct and indirect assays. What is the analytic distribution dilution assays? How are the iterative scheme helpful in for obtaining maximum likelihood estimate? We not explain this maximum likelihood, and direct and indirect assess to I explained. And please read 5 marks question is come in the final paper. Okay, ma? So thank you for giving the opportunity of the BROU team, and especially I thankful to Dr. Vijay Kumar sir for giving this opportunity. Thank you.